Alrighty, so I'm going to be doing my presentation on biofilms. So, microbial biofilms can colonize medical devices and human tissues. They're everywhere and they cause about two-thirds of nosocomial infections. They are also multicellular and have incredible resistance to killing by antimicrobials and host defenses, which means biofilms cause a wide range of infections that are difficult to destroy. And bacterial and fungal biofilms play a huge role in pulmonary and critical care medicine. Most biofilms are surface associated to biotic or abiotic surfaces, and others can be untethered and colonize compromised tissue. Biofilm growth on host tissues and medical devices is now really a key mechanism of virulence for opportunistic pathogens like P. aeruginosa, staphylococcal species, and the fungi Candida albicans and Aspergillus fumigatus. Um, their cells are distinct from planktonic cells with big differences in gene and protein expression patterns. And this mode of growth really refers to the phenotypic traits that enhance microbial survival in chaotic environments and against antimicrobial insults, including drugs and host immunity. And a hallmark of their infections is their incredible resistance to antimicrobial treatments, which leads to difficult relapsing of incurable infections or the need to physically remove infected tissues or medical devices. And with the rise of use of indwelling catheters and medical devices, roughly about 60 to 70% of nosocomial infections are associated with biofilms. So, an important feature of biofilm is the development of the self-produced matrix, so also known as extracellular polymeric substances or EPSs. It surrounds the biofilm cells and provides structural integrity to the microbial community, enhances surface adhesion, cell aggregation, and contributes to antimicrobial and host resistance. The matrix is made of exopolysaccharides, extracellular DNA, and proteins, but its composition varies with growth conditions and microbial species. And I also wanted to add that the hallmark of biofilms is their resistance again to killing or tolerance to many different types of antimicrobials, host defenses, and environmental stress conditions, which allows them to live in hostile environments. They could really live and survive in 100 to 1,000 higher concentrations of antimicrobials or biocides than planktonic cells. Um, and there's many things that contribute to the tolerance, like exopolysaccharides within the biofilm matrix can bind or repel antimicrobials through charge and hydrophobic interactions and limit their intracellular penetration. Um, if the biofilm is altered, it can have profound effects in mitigating antimicrobial activity, and their growth generates microenvironments that create heterogeneous cell populations with different growth and metabolic states or cells under nutrient or oxidative stress. So overall, the biofilm environment facilitates polymicrobial interactions, metabolic functions, and cell-cell communication. And because many biofilms are polymicrobial or microbial interactions that are cooperative or antagonistic, um, may significantly affect microbial virulence, host interactions, and antimicrobial resistance. As far as diagnosis goes, there isn't really a definitive diagnostic marker, so it can be pretty hard to recognize them. They're typically defined by a combination of microbiological, clinical, and microscopic features such as difficult to destroy infections, detection of microbial biofilm on direct exam of the infected tissue, and the growth can also impede diagnosis because microorganisms are not readily recovered for culture without physical disruption of the surface attached biofilms. And sampling of in vivo biofilms is hard, and the traditional diagnostic methods really lack sensitivity and specificity. But susceptibility tests have limited value in predicting activity against the biofilms, and they test the activity of antimicrobials against microorganisms in the planktonic state, not biofilm. And even though they are susceptibility tests for biofilm-grown bacteria, they have not been able to predict microbiological or clinical outcomes when evaluated in patients with CF infected with P. aeruginosa. And susceptibility tests for fungal biofilms have not really been made yet. As far as how biofilms can be a microbial reservoir for nosocomial infections, they're pretty much just everywhere, and they contain the majority of bacteria in their natural habitat. So most water distribution systems are colonized with biofilms, which can contribute to the nosocomial transmission of P. aeruginosa. Surfaces in hospitals can be a reservoir for transmission for pathogens like P. aeruginosa, Acinetobacter baumannii, and Staph aureus, especially in ICU, like where I work. Biocides like chlorhexidine are really ineffective at killing biofilm bacteria, and biofilms have been recovered from sanitized hospital surfaces. As far as anti biofilm strategies, I guess, the antimicrobial drugs have limited success against biofilms, and a lot of research has been done to make treatments that target biofilms by inhibiting their formation, disturbing the established biofilms, or enhancing the activity of conventional antimicrobials. With all the research that has been done, has led to many different surfaces coatings with antimicrobials, and cationic antimicrobial peptides or metal nanoparticles being applied to medical devices like indwelling caths and ET tubes or endotracheal tubes. So, for example, bismuth thiols or silver containing molecules have potent antibiofilm activity. So, also different structural components of the biofilm matrix are also targets for enzyme-based therapies. For example, PLA and PSLG, glycosyl hydrolysis for P. aeruginosa disorder established P. aeruginosa biofilms and enhanced the biofilm susceptibility to antibiotics and neutrophils. And DNA can be an important matrix component of biofilms also. In fungal species, destroying the extracellular DNA with recombinant DNAs weakens the biofilm structural integrity and increases antifungal activity against the biofilms. And then there's very similar observations that are made in bacterial biofilms, but they vary by experimental conditions. Inhaled Dornase alpha is routinely used in CF patients, but mainly for its mucolytic properties. Even though disruption of biofilms can possibly cause dispersed disease, it may be considered in combination with conventional antimicrobial therapy to really target the microorganisms released from the biofilms. Now, really for biofilm-associated infections, there's a bunch of different things. I only chose a few things to really talk about. So there's like indwelling catheters. Catheter infections happen frequently, and they're a complication of using central venous caths in the ICU, which means that, longer length of stay, ICU stay, more cost and mortality. And what happens is the microbial cells stick to abiotic surfaces under static or continuous flow and form surface-attached biofilms that resist host immunity and antimicrobials. Once dispersed from biofilms, organisms will spread to cause bloodstream infections and more colonization. And these infections are usually caused by skin commensal bacterial organisms, although enteric, gram-negative bacilli and opportunistic fungi are encountered in immune-compromised patients or ICU patients. As far as prevention, studies have focused on surfaces covered with antimicrobials or antiseptics to prevent the adhesion and biofilm formation. So, for example, cats covered with chlorhexidine, silver sulfadiazine, or minocycline rifampin have lowered the bacterial colonization of bloodstream infections. And as far as treatment, there's really two ways: disruption of the biofilms, um, and antimicrobial treatment to eliminate viable organisms. So interference of biofilms is very limited to removing colonized cats, and this is suggested because of limited activity of antimicrobials in destroying biofilms. There's also antibiotic lock therapy, which is where we put high concentrations of antibiotic within the catheter to destroy that adherent biofilm. So even though C. albicans biofilms show resistance to echinocandins, these drugs have great activity and they can diffuse catheter-associated biofilms, improve immune recognition and fungal killing, and completely kill catheter-associated infections. And this is facilitated through reduction of beta-1,3-glucan from the biofilm matrix by the inhibitors of B1,3-glucan synthase. And although the clinical efficacy of echinocandins supports their use of treatment of choice for C. albicans biofilm infections, um, current guidelines nonetheless recommend removing the infected cath whenever possible. There's also ET tube colonization and VAPs, or ventilator-associated pneumonias, as most people know in the healthcare field, or in an ICU setting. It's obviously a major nosocomial infection associated with a lot of morbidity, mortality. Biofilms pretty much grow on the surface of the ET tube or endotracheal tube, and bacterial colonization occurs within hours of the intubation. There are many ways that this can happen, like aerosolization of biofilms or mechanical ventilation, disruption during the tracheal sectioning, the bacteria are somehow released and can cause pneumonia. And even though biofilms in the ET tubes aren't sufficient to cause VAP or VAP, they can be a big time reservoir. One article talked about patients who were receiving mechanical ventilation were found to have biofilms, and they did this by scanning electron microscopy in ET tubes. And in 50% of the VAP cases, the same pathogens were identified in the bronchoalveolar lavage fluid and ET tube biofilms. It had to do with treatment failure. Some other studies I found were finding that ET tube biofilms are polymicrobial and made a lot of organisms found within the oropharyngeal and enteric flora, which means that colonization or aspiration of secretions into the subglottic area is a significant route for bacterial colonization of the distal ET tube. There are other studies that have been formed as well, but one thing I found really interesting was one article talked about how silver-coated ET tubes are associated with significantly lower biofilm formation, bacterial lung colonization, and risk of VAP. Other approaches include the ET tube surfaces coated with antiseptics or metal nanoparticles. Even though this all looks very promising, there's still limited data as far as cost effectiveness and safety on these devices. And then one thing I also want to talk about was just kind of the chronic bacterial infections that we see in our cystic patients, or cystic fibrosis patients. And so the thing with these CF patients is that they really have inadequate mucociliary clearance and other impaired host defenses that are caused by mutations in the CF transmembrane conductance regulator gene. And this leads to chronic lung disease characterized by persistent bacterial infections of the airways and destructive lung inflammation. P. aeruginosa is really a big-time pathogen in adult patients with CF, and causes lifelong chronic airway infections that resist eradication by the host immune system and antibiotic therapy. And the main way it does this is by its growth as a biofilm. So it has widespread change in gene expression and with upregulation of exopolysaccharide production, whereas acute virulence genes and motility are downregulated, leading to bacteria that cause less cytotoxicity and invasion of the host cells. It also forms untethered biofilm aggregates within the sputum of CF airways. Bacteria forms similar biofilm aggregates with high density gels when bacterial motility is restricted in conditions associated with CF sputum and chronic inflammation, including the presence of neutrophil elastase. DNA and amino acids can promote biofilm formation. Also, this biofilm growth has resistance to antibiotics and neutrophil killing. And as far as treatment goes for P. aeruginosa, chronic lung infections, it is hampered by biofilm-mediated multi-drug tolerance. And there's also like inhaled antibiotics like tobramycin, which we routinely use in our CF, with our CFers, and are associated with improved pulmonary outcome. Um, Although, despite achieving high pulmonary concentrations, they don't really eradicate the chronic P. aeruginosa infections. But overall, just kind of end things off. These are my references. Um, Biofilm infections are common and cause a lot of clinically significant potential fatal infections. We are still learning a lot more about them and their role in pulmonary infections and how to treat them. And a lot of good studies have been performed, but we still have a long way to go. So thanks for listening.